Hi guys, so today we're talking about the peripheral vascular system. So let's first start off by talking about the anatomy of the vessels in our body. So we have the arteries, the veins, and the capillaries. So starting with arteries. So arteries have three concentric layers. So starting from the inside, we have the tunica intima, and then the tunica media, and then the tunica externa at the very outer portion. Between the tunica intima and the tunica media, we have the internal elastic laminae or the internal elastic membrane. And between the media and the adventitia or the externa, we have the external elastic membrane or the external elastic laminae. Let's first talk about the tunica intima, which is the innermost layer of the artery. So this layer, it's comprised of a single continuous lining of endothelial cells. And although you may think that the structure is quite simple because it's just a single layer of endothelial cells, this layer is actually quite busy. So an intact endothelium, meaning that there's no injury to the endothelium, synthesizes some compounds that help regulate certain processes in the body, like thrombosis, for example, as well as regulating blood flow. So the endothelium can synthesize molecules such as some vasoconstrictors, like angiotensin converting enzymes and endothelin, and vasodilators as well, like nitric oxide and prostacyclin. Also, the endothelial cells in the tunica intima can also help regulate immune response and the inflammatory response through the actions of interleukins and adhesion adhesion molecules. After the tunica intima, we have the internal elastic laminae, and then after that we have the next layer, which is the tunica media. So the tunica media is comprised mainly of, it's comprised of smooth muscle cells that dilate and constrict to regulate blood pressure. And then after that, we have the external elastic laminae, and then after that, we have the tunica adventitia or the tunica externa, which is the outermost layer of the arteries. So the tunica externa is made up of connective tissues containing nerve fibers and the vasa vasorum. So vasa vasorum are they're like small blood vessels that supply the outermost layer of arteries with blood. Now let's talk about arterial branching and the different types of arteries. So there are three types of arteries depending on their size. And the size of the arteries usually vary depending on their distance from the heart. So as we know, the aorta branches straight off from the left ventricle, and then the aorta has like immediate branches following that. So usually these arteries that are near the heart are very, very elastic. So this is actually the first type of arteries. We have elastic arteries. Elastic arteries are the largest and the thickest type of arteries. They contain the most elastin, that's why they're called elastic arteries. And the reason is that so that they could withstand higher pressures because of the heart pumping nearby. These elastic arteries then branch out even further, leading to the more medium-sized arteries, which we call muscular arteries. So this is the second type of arteries. We have muscular arteries. So muscular arteries, they usually deliver blood to a particular organ. So examples of these include the femoral artery, the mesenteric artery, and even the renal arteries. So these are different from elastic arteries because they contain more smooth muscle cells in their tunica media compared to the elastic arteries. And then these branch out even further to smaller and smaller arteries called arterioles. These arterioles then lead to capillary beds. So capillary beds are just network of capillaries. So capillaries are the smallest type of blood vessels. So capillaries are just made up of a single lining of endothelial cells. So they just have the tunica intima. It's coated in a thin layer of basement membrane, but they don't have any tunica media, don't have any tunica adventitia. Literally just one single lining of endothelial cells. And sometimes you can get these cells called pericytes that wrap themselves around the capillary wall. These pericytes, they're embedded in the basement membrane, and they function just to stabilize the capillary walls. The lumen of capillaries are very, very small, sometimes just small enough to let red blood cells through in one single file. So let's review what we've discussed just now. So we have arteries, and arteries, they have three layers: tunica intima, tunica media, and tunica adventitia. And there's three different types. We have elastic arteries, the biggest ones, and then we have muscular arteries. Then they branch off into arterioles, leading into capillary beds, and we have this teeny tiny capillaries. And then at the other end of that, we have venules branching out into the bigger and bigger veins. So veins, in comparison with arteries, veins have thinner walls, and they have less smooth muscle in the tunica media compared to arteries. And this is because they experience less pressure. And veins, they have valves to prevent the backflow of the blood, especially in the limbs where blood has to go against gravity. So the valves are there to prevent backflow and to help direct the blood back up to the heart. The veins from the arms, the head, the neck, and the upper trunk, they all drain into the superior vena cava. And the veins from the legs, the abdomen, and the lower trunk, they all drain into the inferior vena cava. And all of this blood goes straight to the right atrium. So in the legs, actually, we have deep and superficial veins. So the deep veins, they carry approximately 90% of venous return from the lower extremities. And these veins are very well supported by the tissues around them. In contrast with the superficial veins, because the superficial veins, they are subcutaneous veins. So they have relatively poor tissue support compared to the deep veins. Superficial veins include the great saphenous vein. So the great saphenous vein joins the deep venous system via the femoral vein. And then we also have the small saphenous vein. And the small saphenous vein joins the deep venous system in the popliteal fossa. Also in our legs, so in addition to the valves being present in the veins to prevent backflow and direct blood to the heart, in the legs we also have the contraction of the calf muscles when we're walking. So the contraction of calf muscles when we're walking kind of acts like a venous pump, propelling blood back up to the heart against gravity. Let's quickly rewind and talk about arteries again, because I forgot to mention arterial pulses. So you know that we can't palpate every single artery in the body because some of them, they lie deep within our bodies. So arterial pulses are palpable in arteries that lie close to the surface of the body, close to our skin. So there are different arteries in our body where we could feel the pulsations. So we could feel them in our arms, in the abdomen, and in our legs. So in the arms and hands, we have the brachial artery, we have the radial artery, and we have the ulnar artery. And actually, there's two vascular arches that connect the radial and the ulnar arteries together. So, and that exists to protect the circulation in our hand in case of a possible arterial occlusion. And the arteries in the abdomen, so we can palpate the aorta in the epigastrium, but the three deeper branches of the aorta we can't really palpate. So we have the celiac trunk, we have the inferior and the superior mesenteric arteries. The celiac artery supplies blood to the foregut. So that includes the stomach, the liver, the pancreas, the spleen, and the gallbladder. The superior mesenteric artery supplies blood to the midgut. So that includes the jejunum, the ileum, the cecum, the large intestine, the ascending and the transverse colon. The inferior mesenteric artery supplies blood to the hindgut. So that includes the descending and the sigmoid colon, as well as the proximal rectum. The arterial pulses in our legs include the femoral artery. I don't know what I was going to show you, but the femoral it can be palpated between the anterior superior iliac spine and the pubic symphysis. We also have the popliteal artery just behind the knee. We also have the dorsalis pedis and the posterior tibial artery. Now let's discuss the transcapillary exchange, or the exchange of substances in and out of the capillaries. So as we know that the left ventricle pumps oxygenated blood, which means blood with oxygen and other nutrients, through the aorta, which then splits into its branches, which then splits into other branches until it gets to the arterioles, until it gets to the capillary bed. So in the capillary bed, we have capillaries, and capillaries are very porous. So they have pores which allow substances to leak out and leak in. So to be pushed out and to be pulled back in. So I drew this diagram to help me explain what's going on. Sorry guys, let's continue. So as I was saying, I drew a diagram to help me explain this concept of transcapillary exchange. So here we have a single capillary, and the capillary has two sides. So we have the arterial side here in pink and the venous side here in green. So as we know that the capillary, it's not like a continuous tube, it's actually porous. So it has gaps in the membrane, which means that stuff are gonna be leaking out and coming back in. So stuff are allowed to go in and come back in, to go out, sorry, and come back in to the capillary. So, right. So we know that when the heart pumps blood, right, it pumps blood under a certain amount of pressure. So ideally, our blood pressure should be 120/80, but here we're talking about the mean arterial pressure. So what is the mean pressure that the heart, the blood exerts to the walls of the capillaries as it goes through the arteries? So to calculate arterial pressure, we need to take the diastolic blood pressure plus a third of the pulse pressure. So the diastolic blood pressure is 80. And the pulse pressure is the difference between the systolic and the diastolic blood pressure. So that is 40. So 120 minus 80 is 40. And a third of that is around about 13. So 13 plus 80 is 93. So the mean arterial blood pressure is about 93 millimeters of mercury. So those arteries branch out into smaller and smaller and smaller vessels, into arterioles and into capillaries. Due to that extensive branching, when we get to the capillary, the arterial end of the capillary, the blood pressure is reduced so much from 93 to around about 30 millimeters of mercury. It's around about 30 to 35 millimeters of mercury, but let's stick to 30 millimeters of mercury in this example. So again, at the arterial end, blood is exerting 30 millimeters of mercury pressure against the wall of the capillary. Because it's exerting that pressure and the capillary is porous, stuff are going to be pushed out of the capillary through those gaps. So let's just quickly review what's actually inside the blood. So inside the blood, we have different components. So we have water, obviously. We have red blood cells. We have white blood cells. We have platelets. We have some proteins. So the most abundant protein in the blood is albumin. We also have like electrolytes, glucose, oxygen, carbon dioxide, so nutrients, waste, gases. So these pores, these holes are too small to let any of these guys pass through. So it's not going to let red blood cells, white blood cells, platelets, and proteins pass through because, because the gaps are too small, so they're not going to fit. So when stuff gets pushed out of the capillary in the arterial end, these guys, they stay in. Okay. So what stuff actually gets pushed out? So oxygen diffuses out of the capillary anyway, so it reaches the tissues. Glucose also gets pushed out. What else? So fluids and certain electrolytes gets pushed out. But because these guys are still here, because of osmosis, so recall that osmosis is the movement of water through a selectively permeable membrane. So here we have a selectively permeable membrane, which is the capillary. And these blood components here, especially the protein, especially albumin, it exerts a pulling force because of osmosis, okay? So because water would want to balance the concentration out, these components, these blood components, exert a pulling force. So they pull stuff back in, including water, okay? So that pulling in force, that pulling in pressure, has a value as well. So that's 20 millimeters of mercury, okay? So here in the arterial end, we see that 30 millimeters of mercury pressure pushes stuff out of the capillaries, and 20 millimeters of pressure pulls stuff back into the capillaries. So that means that we have a net filtration of 10 millimeters of mercury. So we have a net pressure, so 30 minus 20, 10, and the direction is outside because the pushing in force, the pushing in pressure is greater than the pulling in pressure. So in this end, we get filtration. We get stuff being pushed out of the capillary. As the blood components move through the, the capillary, we reach the venous end of the capillary. So at the venous end of the capillary, because we're slightly further away, and you know, when you increase the distance from the source, the pressure decreases and decreases. So at this end, the push out pressure is now reduced to 15 millimeters of mercury. So we started at 30 at this end, and here we got to about 15 millimeters of mercury. So the pushing out force is not as great as the pushing in force in this end, okay? But the pulling in force remains the same because these same components, because they're not being pushed out, they just keep inside that tube until it gets to this end. So they're still going to exert around about the same amount of pressure, which is 20 millimeters of mercury. So this means that the pushing in force is greater than the pushing out force. So the net force here is 5 millimeters of mercury, and the direction is towards the capillary. So at the venous end, we get stuff being pulled back into the capillary. So we get like waste products here, we get carbon dioxide diffuses, so that's fine. So we get like waste products and fluids being pulled back into the capillary in the venous end. So this is perfect because it allows us to deliver nutrients and oxygen to the tissues at the arterial end. And when we get to this end, it allows us to collect the waste products and CO2, which gets then delivered back up to the vena cava and back up to the heart and then out into the lungs. So this is actually a really perfect system. So this pushing out and pulling in pressures that I was talking to you about, they actually have names. So in pink, we have the pushing out pressure. So that's called the hydrostatic pressure. It's basically the blood pressure because it's that pressure that generates this pressure inside the capillary that pushes stuff out. And then in green, we have the colloid osmotic pressure. So this is the pulling pressure exerted by these blood components, in particular the proteins, specifically albumin. Actually, it exerts a really high pulling force there, pulling pressure. So we have two opposing forces. We have the hydrostatic pressure pushing stuff out, and we have the colloid osmotic pressure trying to pull stuff back in. And in this arterial end, the hydrostatic pressure dominates, so we get a net filtration. So that means we get stuff being pushed out. And in the venous end, the colloid osmotic pressure is greater than the hydrostatic pressure, which means that we get a net reabsorption. So we get stuff being pulled back into our capillary. So I hope that makes sense. I want to use this diagram as well to explain some of the mechanisms for edema, because edema is one of the most common concerning symptoms in the peripheral vascular system. So there are different ways in which edema can occur. So there are different mechanisms. So one of them include increase in plasma volume due to sodium retention. It can happen because of altered capillary dynamics, which means that for some reason, we have more filtration and less reabsorption. It can also be due to not the capillary themselves, but due to another system, the lymphatic system. So it can be due to inadequate removal of fluids from the lymphatic system, or an obstruction in the lymphatic system. And the last one is the increase in the size of pores in these capillaries. So I want to explain how that works and how that will lead to edema, and then we'll discuss the lymphatic system for completion. So all of this will tie in together and it will make perfect sense, I promise you. Right, so let's discuss let's discuss one of the mechanisms of edema, which is the increase in capillary permeability. So if we increase permeability in the capillary, which means that we make these pores bigger, what's gonna happen is that some of these components of the blood will be able to now leak out of the capillary. In particular, our proteins, our albumin and our proteins. So when they leak out of the capillary, what's that gonna do? That means that we're not gonna have much pulling in force anymore. So we're just gonna have lots of like fluid and like stuff being pushed out. But because we lost that component that establishes or that actually generates that pulling in force or pulling in pressure, then that means we're just gonna keep, filtering stuff out into the interstitium. So this is the tissue, this is the capillary, and the space in between is the interstitial space or the interstitium. So fluid will just accumulate here. They're not much of them are gonna be reabsorbed, because those components, those molecules, those compounds that generate that pulling in force are now outside of the capillary. So we're just gonna end up with lots and lots of stuff being pushed out, especially fluids, and they're just gonna be here in the interstitium, and not much of that fluid is gonna be reabsorbed back, as we have just decreased the colloid osmotic pressure by a lot by losing those proteins. Okay, so I hope that makes sense. Now let's move on into talking about the lymphatic system. The lymphatic system is an extensive vascular network that just drains the fluids from the tissues and bring them back into the venous circulation. That's all that it does. So let's define some certain concepts. So lymph, let's start with lymph. So lymph in Latin, here we go with my Latin again. Lymph in Latin means clear fluid. So lymph is the clear fluid that flows through the lymphatic vessels and the lymph nodes. So the lymphatic vessels, the lymph nodes, and the lymph, they make up the lymphatic system. So the functions of lymphatic system, well, I've mentioned one already, which is to return fluid that accumulates in the tissues back to the venous circulation and therefore back to the heart. So in this diagram, the lymphatic system is actually missing, but in real life, it's there, trust me. So the tissues are here, and when stuff gets pushed out, including fluids, some of them obviously gets absorbed into the tissues and in the interstitium as well. So those fluid, they get collected by the lymphatic system and returned back into the circulation. The second function of the lymphatic system is to help deliver big molecules into the circulation. So for example, lipids and hormones. The third function of the lymphatic system, which is very important, is immune surveillance. As a fun fact, actually 20 liters of fluid seeps out of the capillaries, but 17 liters of that 20 liters actually gets reabsorbed quickly back into the capillaries. So there's 3 liters that just seeps out into the interstitium and into the tissues, and it's the lymphatic system that brings that fluid back into the circulation. And once that fluid is inside the lymphatic vessels, it's not called blood, it's not called fluid, it's called the lymph. Okay, so I hope that made sense. So in the interstitium we have lymphatic capillaries. So these are the smallest of all the lymphatic vessels. These lymphatic capillaries are very permeable. So they are made up of endothelial cells that loosely overlap. So they have like really big gaps in between the cells. This makes them extremely permeable. So these endothelial cells are anchored into the interstitial space by collagen, and this allows them to be flexible but also to return its shape. So the fluid collected in the lymphatic capillary, which we now call as lymph, travels through the lymphatic system. So it goes through the lymphatic vessels, into the lymphatic trunks, into the collecting ducts, and eventually being dumped into the venous circulation through the big veins in the neck. So there's two main lymphatic ducts. So we have the right lymphatic duct, which drains the right side of the head, right side of the neck, right side of the upper thorax, and right upper limb. And the other one, the thoracic duct, kind of drains the rest of the body. So it's actually a much bigger duct. The right lymphatic duct dumps its lymph back into the venous circulation through the right subclavian and the right jugular vein, whereas the thoracic duct dumps its lymph into the left subclavian and left jugular vein. Similar to the veins, there's no pump in the lymphatic system. So the lymph vessels, they just kind of rely to the pulsating arteries nearby and the squeezing or the contracting of the skeletal muscles nearby to help propel the lymph back into the venous circulation, back to the lymphatic trunks and the collecting ducts, and then into the venous circulation. The lymphatic trunks, they have valves which direct the flow of lymph back to the collecting ducts and into the venous circulation. And like I said at the beginning, the lymphatic system is not just to collect the extra fluid in the interstitium and in the tissues and bring it back to the venous circulation. They actually have immune functions, specifically for immune surveillance. So certain lymph organs actually remove foreign materials from that lymph, so that those materials or those substances or compounds don't enter our bloodstream. So an example of a lymphoid organ is lymph nodes. Lymph nodes are round, oval, or bean-shaped structures. They're basically just consist of lymphoid cells plus some proteins, and they vary in size depending on their location. And if they are concentrated along a lymphatic trunk, then they may be able, then we might be able to palpate these. Only superficial lymph nodes can actually be palpated. So what actually happens is, as the fluid goes through the lymphatic system, it will pass through one of these lymph nodes. So one of these collection of lymphoid cells. So in the lymph node, we have a specific type of cell called dendritic cell. So dendritic cells are antigen presenting cells. So what they do is they constantly sample things and present bits of that into like other cells called the B cells. So if our dendritic cells break up pathogens or any like foreign materials or substances, the B cells would then recognize that as something that's harmful or a threat, and therefore that B cell would would turn into plasma cells. So that plasma cell will now then produce antibodies that would neutralize this pathogen. The axillary lymph nodes drain most of the arm, but some gets drained directly into the infraclavicular nodes. In the lower extremities, so in the legs, the lymph nodes, they follow the venous system. So we have the deep and the superficial venous, the veins. So we have deep and superficial nodes as well. I'm just gonna mention some of these lymph nodes really quickly because the book talks about these and they might be in the exam. So the superficial inguinal lymph nodes, they accompany the great saphenous vein, so one of the superficial veins of the leg, and they are grouped into two main groups. So we have the horizontal group that lie they lie on the anterior thigh, just below the inguinal ligament. And we have the vertical group that lie at the upper part of the great saphenous vein. So the lymphatics accompanying the small saphenous vein, which is the other superficial vein, joins the deep system at the level of the popliteal space. That means that the lymphatics accompanying the small saphenous vein drain into the popliteal lymph and then into the deep inguinal lymph nodes. And the lymphatics accompanying the deep veins in the leg, like the femoral and the popliteal, they drain into the popliteal lymph and into the deep inguinal lymph and then into the deep iliac lymph nodes. For the final part of this video, I just want to quickly talk to you about atherosclerosis and the formation of atheroma. So atherosclerosis is a chronic inflammatory condition that is initiated by the injury in the endothelial cells in the tunica intima, the innermost layer, provoking an atheromatous plaque formation. The atheroma, it starts, as I said, with the injury in the endothelial cell in the tunica intima. So once we have that injury in the tunica intima, it attracts circulating lipoproteins, specifically the LDLs, the low-density lipoproteins. So once those low-density lipoproteins get into that injury site, they initiate an immune response or an inflammatory response. So this inflammatory response attracts monocytes, and these monocytes, once they get in the tunica intima, they mature into macrophages. These macrophages eat or feed or engulf these lipoproteins, and they become foam cells. When these foam cells die and degenerate, they release their intracellular contents into the extracellular space, forming an extracellular lipid pool we then call an atheroma. And over time, fibrous tissue accumulates and a hard plaque forms over this atheroma.